Let's move on to the next oral presentation entitled Identification of FAM111B as a potential therapeutic target for bladder cancer, presented by Lei Peng.
Okay, honorable judges, my name is Lei Peng from Naval Medical University. It's my great honor to introduce our project, Identification of FAM as a potential therapeutic target for bladder cancer. My presentation covers the following 5 aspects. Bladder cancer, also short for BRC, is the most prevalent malignant tumor of urinary system. There are about 550,000 new cases each year in the world, and the mortality rate is as high as 33%. The survival rate of patients could be improved by surgery and chemotherapy. Unfortunately, the onset of bladder cancer is occult, and the early symptoms are not obvious, which are usually found in the middle and late stages, causing unsatisfactory prognosis. Thus, effective therapeutic targets are urgently needed. FAM belongs to family with sequence similarity 111, contains a predicted trypsin-like cysteine or serum peptidostigmine. It has been reported to be associated with several types of human cancers. For example, it could enhance proliferation of KRAS-driven lung adenocarcinoma by degrading P16 or as a biomarker for hepatocellular carcinoma. Exploring the function of FAM in BRC could lay a solid foundation for targeted treatment for BRC therapy. Based on this data, our research mainly focuses on 3 questions. Is FAM related to the occurrence of bladder cancer? What's the role of FAM in progression of bladder cancer? Could FAM be a therapeutic target for bladder cancer? Here's our technical route. First, we're collecting samples from patients with BRC and conducted immunohistology and clinical statistic analysis. Second, we use the cell models with FAM gene knockdown to perform proliferation, migration, and apoptosis experiments. Xenograft mice were used to conduct tumor formation study. Finally, we use microarray data to search for possible molecular mechanisms. Here are our results and discussion. As shown in figure 1 and 2, the majority of tumor tissues have stronger FAM immunostaining than that of tumor tissues. We divided all patients into 2 groups according to median IT score and found that patients with high FAM expression were more likely to suffer from lymphatic metastasis. PM survival analysis was performed utilizing TCG database. The results indicate hyperexpression of FAM was related to short disease-free survival. In conclusion, the expression of FAM is correlated to BRC progression. Examine the biological function of FAM in BRC motility. We knock down FAM in T24 and EG cell line by lentiviral transduction. They are the 2 human BRC cell lines, and this was the most efficient one. qPCR and Western blot also confirmed the FAM1 could knock down FAM most effectively. Cell counts were used to determine cell proliferation complexity. The results show the 2 cell lines in FAM group exhibited slower proliferation rate. With healing assay showed, in T24 cells, the migration rate of cells was decreased by 50%, while there wasn't significantly different in EG cells. In Tresvell experiment, compared with migration rate of T24 cell line that was remarkably inhibited, there wasn't significant difference among EG cell lines. These two results demonstrate that knockdown FAM only influence the migration rate in certain BRC cell lines. FAM's effective migration in cell line may not be the main influencing factor. Flow cytometry was utilized to estimate the proportion of apoptotic cells, and the proportion increased significantly after knockdown of FAM. In conclusion, knockdown of FAM inhibits cell proliferation, migration, and induces cell apoptosis in vitro. Moreover, reconstructed xenograft models by implanting P24 cells perfected with FAM or control groups into immunocompromised mouse to assess the value of FAM in terms of BRCA growth in vivo. After 30 days, only half of xenograft mice injected with FAM knockdown cells formed tumors, and the tumor sizes were smaller. In vivo bioluminescence imaging also confirmed heavy tumor burden in control group, and tumors in control group grew faster during cultivation. We further carried out Ki67 staining, and the expression of Ki67 shows significant decrements, which suggested the weakening of cell proliferation ability. In conclusion, knockdown of FAM suppresses BRCA growth in vivo. Finally, we use microdata for transcriptome profiling and IPA to explore the molecular mechanisms by which FAM contributed to BRCA growth. It was suggested that reduction of FAM expression level could activate AMPK signaling and suppress CDC42 and REX signaling pathway, which might be the direct pathway for the inhibition of BRCA growth. In summary, FAM was found to mainly promote tumorigenesis, progression of BRCA, which could be a promising target for BRCA therapy. In the future, we will not only clarify the direct pathway of FAM in the tumorigenesis, progression, and metastasis of BRCA by IPA, qPCR, and Western blot, but also further explore the downstream targets of FAM by co-IP, which will provide the theoretical basis for targeted treatment of BRCA. The purpose of basic research is to explore molecular mechanisms to better serve clinical drug development. We sincerely hope that our research can make a contribution to this goal. Thank you for listening.
What a magnificent experience that is.
Do you have any comments or any questions? Okay.
So thank you for your presentation. I'm wondering about the cell line experiment that you have performed. So you have chosen the T24 and EJ cell line, and some data showing that there are differences between the two in the effect after the knockdown of the gene of interest. So what is the mechanism and what are the differences between these two cell lines?
Thank you for the question. And speculate that it's the difference between T24 and the EJ cell line, and T24 is a metastasis cell line of bladder cancer, while EJ is an in situ cell line of bladder cancer. Thus, the migratory ability of EJ may be weaker than T24.
Sorry, they are not the bladder cancer cell line, both of them?
No, they are all the bladder cancer cell line, but the T24 cell line is a metastatic cell line of bladder cancer, while EJ is an in situ cell line of bladder cancer.
Okay.
And,
So maybe one of them is more potential to be metastasis. So can you, if you look into literature, is there any explanation that why one of the gene, the knockdown of the gene is working in one cell line and is not in the other? Because maybe it can explain the mechanism.
And we also speculate that the BRCA, the effect that BRCA2 cell lines to migration rate may not be the main influencing factor, and the development and the prognosis is the main factor of FAM gene.
Thank you so much.